Swayam Prabha. Digital India. Educated India.
Welcome to today's lecture. This is lecture number 20. We are going to continue about the MHC, the major histocompatibility complex, because in last lecture we are studying the last slide was about the size of peptide which MHC 1 and 2 can accommodate. I am just reminding you again, the MHC 1 can accommodate 8 to 10, or in some books you will find 9 to 11, and MHC 2 can accommodate bigger and more wide variety of peptides, and that is 13 to 20. Okay. So, we already know why this peptide is important, because these peptide which is actually processed and presented by MHC are going to recognized by T cell receptor. We are talking about the B cell epitope. You know by now what is epitope. Epitope means the segment of the antigen which is recognized by B cell receptor is known as B cell epitope, and the segment of the antigen which is recognized by T cell receptor is called T cell epitope. So, for a particular antigen, that T cell epitope and B cell epitope may be same. Okay. So, the same antigen may be recognized by B cell and recognized by T cell, but in many times or several times what happened, the B cell epitope is not exactly the same which is T cell epitope, because B cell epitope always should be at the surface of the antigen or the protein which is recognized. So, it should be on the surface. So, the antibody can recognize it, but in case of T cell what happened, this whole peptide will be open and chopped into pieces. Suppose this whole hand is the antigen, it will be chopped into different pieces. So, all the fingers will be separated. One of this finger after processing will be represented by MHC, either 1 or 2. So, that may be buried inside the cell, inside the antigen. Okay. So, it not necessarily it should be on the surface of the antigen. So, there is a difference, but definitely the surface and surface epitope also may be presented by T cell. So, the size is different. MHC 1 is smaller. I mean in presentation the peptide that MHC 1 is presenting is small with respect to MHC 2. So, today before going to discuss about the MHC, what we will discuss, like we have a multiple copy of MHC. Okay. So, every individual have a different copies of MHC 1 and MHC 2, and MHC genes are one of the most polymorphic gene so far discovered, maybe the most polymorphic gene so far discovered. So, why this polymorphism and more number of copies required? Not only that, because these presentation or the antigen binding cleft is not as specific as T cell receptor or B cell receptor, right, but still we need so many number for several reasons. One of the most important reason is that multiple MHC can present multiple different type of peptides after processing. This is number one. Number two is, if there is only 1 or 2, say suppose there is only one MHC 1 and one MHC 2 which can represent all possible peptides, then what will be the problem? Just in case if there is a mutation in that MHC or a problem in that MHC, whole immune system will be collapsed, because whole T cell activation will be collapsed. So, that is one reason that one copy is always bad for this kind of protective system. Second is, there are many viruses which can manipulate the MHC. Okay, they can somehow block the MHC function. So, if that is the case, then if there is only one MHC, if by any chance it is blocked, not mutated or not deleted, so that means we are not inhibited, but viral infection if it can stop, what is going to happen is then whole immune system will be blocked again. So, if we have a multiple copy, so one virus can block 1, 2, but still we will have enough in number, so that our immune system is not completely blocked or locked down. So, we will have the immunity. Okay. And polymorphism is definitely very important, because until unless we have a multiple, this polymorphic situation, we cannot have many variety of MHC, and different MHC can present different variety of antigen, which will make the immune system more diverse. So, more T cell can be accommodated in to involve in the immune system. Okay. So, let us go to the slide just to see again we are going to discuss about our immune system. So, we discussed about MHC presented antigen and MHC and the TCR interacting, but how they are interacting. The crystal structure actually tell all about this. The crystal structure of several peptide, MHC complex and T cell receptor. So, MHC with peptide and T cell receptor complex are crystallized, and the structure is saying that similar orientation. What I mean by similar orientation? That means, so if this is MHC 1, okay, this is a crystal structure, this is MHC 1, let us go to next slide. This is MHC 1. If you see, this is alpha 1 domain, alpha 2 which is the peptide binding region, the yellow one is the peptide, and alpha 3 domain, and this is beta to M, that means beta microglobulin. Okay. So, these alpha 1 and alpha 2, if you see, this is the peptide and this is the T cell receptor. You see V beta, because T cell receptor MHC molecule, the T cell receptor has one alpha chain, another beta chain. So, the alpha 1 of MHC is interacting with beta chain variable region, and alpha 2 is interacting with variable region of alpha chain of T cell receptor. So, beta alpha 1, and so this is not only one. So, several MHC peptide TCR complex crystal structure has been solved, and most of the cases it was found that the crystal structure shows that the interaction is between alpha 1 of MHC 1 with beta variable region and alpha 2 of MHC 1 with beta alpha variable region. Same way, when we see the MHC 2 TCR complex, similar, like this is the beta 1 beta 2 of MHC 2 and this is the alpha 1 alpha 2 of MHC 2. You can always remember alpha beta. Okay. So, why I am saying alpha beta? Because you see the MHC 2 alpha 1 is interacting with variable beta. So, alpha beta and alpha beta. Okay. So, this alpha variable region is interacting with beta 1 domain of MHC 2 and beta variable region is interacting with alpha 1. So, it is very easy to remember, okay, that this is MHC molecule, this is peptide. Definitely the internal part, I told you in a figure with a figure, that internal part, and if you just correlate with the antigen, if you correlate with the antigen, so if this is, say, let me draw, if this is MHC, like the way we used to see, if this is MHC, so this is a membrane, and if this is the peptide, okay, this is peptide. Now, if I use the TCR is green, this is TCR. You see this part is interacting. So, TCR this part, if it is, let me put a line, this is like this is V beta and this is V alpha, and it will be alpha 1 and this will be beta 1. So, this I am comparing with this. So, this domain is interacting with alpha and beta, and the center part of the T cell receptor is actually interacting with the antigen. Okay. So, that way the alpha, in in TCR that we will see in T cell receptor also, the part interacting with the MHC is not that much variable, and the central part of the TCR is contributed by what? I told you before also, this is the CDR3. So, CDR3, that is why most variable in T cell receptor, in antibody also, because they are interacting with variety of antigens. Though there are variety of MHC, but it is not as much as different variety of antigen. So, it is not going to be as much as many variety of antigen, because MHC has a limited number, antigen is unlimited, right. So, this MHC definitely interacting with the TCR, MHC with antigen is definitely interacting with the TCR, but along with that it is also interacting with the CD4 and CD8 surface protein. We already discussed about CD4 and CD8 as a co-receptor of T cell while we are discussing the varieties of T cell receptors. So, it is also interacting with CD4 and CD8. If you remember, I told that CD4 present in the helper cell and CD8 present in the cytotoxic T cell. How it is determined that, and if I ask you a question like which kind of antigen is presented by MHC1? MHC1 is presenting the intracellular antigen, that means the protein synthesized inside the cell is presented by MHC1, and that is recognized by cytotoxic T cell. So, that cytotoxic T cell is going to what? Is going to do? They are going to kill the cell. So, cytotoxic T cell is recognizing either virus infected cell or tumor cells. In both the cases, the protein is synthesized inside the cell, and that is presented by MHC1. In that case, it should be recognized by cytotoxic T cell and cell should be killed. But in case of MHC2, it is mostly helping or mostly presenting the T helper cells, because that T helper cells are going to get the signal from MHC TCR complex, antigen complex, and that signal will ultimately help the B cell to proliferate. So, anything coming from outside is going to present by MHC2, and MHC2 is interacting with CD4 cells, that means helper cells. So, how it is determined actually? If you see, this is this picture, this slide you already have seen, this slide the CD4 and CD8 you know, I am not going to go detail. But if you see this picture, you see the MHC class 2 molecule, alpha beta is interacting with cytotoxic, I mean with T cell receptor along with the peptide, but it is also interacting with the CD4. Okay. So, this interaction is interaction, that means the beta chain of MHC class 2 are very specifically interacting with CD4. Similarly, the alpha 2 and alpha 3 domain of MHC class 1 is interacting with CD8. So, just this MHC1 and CD8 cell, that means the cytotoxic T cell interaction was also not only made sure, because if this cross react, that will be a serious problem in the immune system. Suppose suddenly the CD8 started interacting with MHC2, okay, CD8 started interacting with MHC2, that will be a serious problem. You can imagine, I will tell, but this is also, this is the bigger version, that MHC interact with TCR as well as MHC2 interact with TCR as well as it interacting with CD4 co-receptor of the T cell. Similarly, CD8 interact with MHC class 1, because this interaction is necessary. This interaction is necessary because cytotoxic T cell recognition is not a good thing. So, if by mistake if it recognize any of our own cell, that will kill our own cell. So, that mistake should not happen. Okay. So, only virus infected cell and tumor cells should be recognized by the cytotoxic T cell, otherwise it will be a big problem if there is any cross matching, like if MHC2 is recognized by the CD8 8 cells. Okay. This is kind of, I am repeating the same thing this way or that way, may be little confusing, but it is very simple. I already once told, it is very simple and straightforward. Will come when antigen presentation will come, that time it will be very much clear why this thing happened, though it is discussed in very early classes. So, this is the crystal structure where it is showing that alpha beta T cell receptor, MHC class 2, CD4 all interacting. It is not just, I mean, why I am showing this picture, you do not have to remember all this thing, but it is not just the cartoon I draw that is. So, when it is crystallized, it is also found that the CD4 is interacting with this MHC class 2 and definitely with the TCR molecule of alpha beta type. Okay. So, this is to prove or the confirmation that whatever in the previous slide was shown is not just people thought that this thing is happening. It is happening and it is proved by crystallization of the whole complex. This is the same thing with CD8 like TCR, MHC1 and CD8. In both cases, it is not just a speculation, it is fact. Okay. So, these two classes, now I mean now it will be very clear what I was discussing before. These two classes of MHC, that means MHC1 and MHC2 are expressed differently on cells. What that means differently on cells? I will show you a table. See, if you see, this is MHC class 1. Okay. So, this column is MHC class 1, this column is MHC class 2. If you see T cell express MHC class 1 heavily, MHC class 2 is not that much. Okay. Why? Because in human cells, activated T cells express MHC2, okay, whereas in mice, I mean where it is tested or most of the immunology experiment was taken, done, in that case mouse is completely MHC class 2 negative, which is also written, I kept it written just in case if you forget this, what does this asterisk means. B cell express both of them highly positive, that means they are expressing a lot. Macrophage expressing MHC1 very much, MHC2 little less, but they are expressing a very good amount. Dendritic cells, they are also expressing both MHC1 and MHC2 heavily. Epithelial cell of thymus, MHC1 is very little, MHC2 is very high. If you go to other nucleated cells, so what is the similarity of these cells? Because they are all immune cells or cells which are involved in immune system, but other cells like normal cells, neutrophil is definitely is a part of immune system, they express mostly MHC class 1, not MHC. So, other cells if you see, except this top 5, neutrophil, hepatocytes, kidney, brain, or you take any somatic cells which present in other part like skin, you won't see they are expressing MHC class 2. Okay. But all of them, neutrophil is definitely expressing good amount of MHC class 1, but other cells all are expressing MHC 1. If you take any cells from body, most likely they are expressing or all of them are expressing MHC 1. Okay. Maybe their expression level varies between each other, but MHC 2 is very much restricted to very few cells. Okay. If I ignore the T cells, because it is only in the activated T cells sometimes express that, B cells, macrophage and dendrite and epithelial cell of thymus, which is not normally we need it, because thymus is only responsible for the development of T cells, and after puberty thymus is disappeared. Okay. So, in any adult individual we do not have thymus anymore. So, this thing if we just ignore for the time being, then B cell, macrophage and dendritic cells, these 3 cells are express the MHC2 at maximum level. Okay. Rest almost no or 0. Why? Why this thing happening? What is the role of this B cell, macrophage and dendritic cells? B cell, macrophage and dendritic cells are antigen presenting cells. They are professional antigen presenting cells. They have the responsibility or they have the duty to process the antigen and present through MHC2 to T helper cells, and which in turn help the B cells to produce antibody. So, major contribution of adaptive immunity or antibody mediated immunity, you need that antigen presenting cells. So, antigen presenting cells are B cells, dendritic cells and macrophages. They are the only 3 antigen presenting cells. We call it professional antigen presenting cells. They need MHC2 because they need to present the process peptide to T helper cells. Rest of the cells they do not need that because they do not have that responsibility, they do not interact with the TCR. That is why we do not have. Okay? Any cells does not have this MHC2. One thing we should remember that every cells in our body, they are very economic. Economic in that sense, they do not express any protein if they do not need, they do not waste any single ATP if they do not need. Okay? So, they are very particular about it. So, if any cell does not require to present the antigen, they do not need MHC2, but why we need MHC1? Because MHC1, if you go back to the lecture, early part of this lecture, what I told is MHC1 is responsible to present internal peptide or internal protein, in endogenous protein more specifically. That means, the proteins those are synthesized inside the cell are going to be presented by MHC1. So, all virus infection proteins are expressed inside the cell. If any cell converted to tumor or cancer cell, they express the protein inside the cell. Okay. So, if you think carefully that any part of our body is very much prone to infected with virus, like say for example, if I say the hepatocyte, the liver cells, any hepatocytes can be infected by hepatitis virus, hepatitis B, C, right. So, any cell of our body can be infected to by virus, or any part or any tissue of our body can convert to cancer cell or can convert to tumor cells. So, immune cell need to protect only by cytotoxic T cell to kill them. So, all cells need MHC1 because all cells of our body can be infected by virus. Not only that, any part of our body can be converted to or transformed to cancer cells. So, to protect that, all cells are expressing MHC1 because all cells need it, but MHC2 we do not need it, and that is why it is like distributed automatically by evolution that way. Wherever we do not need, it is not there. If you see the bottom part, the red blood cells, neither MHC1 nor MHC2, because RBC, you know, human RBC particularly, we do not have nucleus. So, if there is no nucleus, no protein synthesis. So, if there is no protein synthesis, MHC1 is not required. If there is no protein synthesis, MHC1 is not required. And they cannot eat or do phagocytosis, or they cannot synthesize the MHC2 also. So, it is not there. Okay. So, this is the general distribution of MHC1 and MHC2, and it is also, I mean, if you remember this, automatically you can remember their function, like what MHC1 and MHC2 do, or either way. If you remember what is the role of MHC1, you can remember their distribution in a different part of our body or different cell system of our body. Okay. So, now I am just seeing the red blood cells. I can't stop myself to tell you, like if you remember different pathogens, like our immune system can handle them. If you remember, and most of you know, I am sure, I am just repeating it again, and those who do not know, just for them, malaria parasite, Plasmodium, the Plasmodium falciparum mostly, there are vivax and others, but Plasmodium falciparum, you know, they are growing inside, you know, right, they are growing inside the RBC. Okay. Imagine how smart they are. So, first thing, they grow inside a cell. So, immune system will not see them. Not only that, that RBC where they are growing and killing one by one and they replicate inside the cell, they can't express, or they do not express, they do not have the machinery to express MHC1 and MHC2. So, while they are growing inside, if you remember the mycobacterium which grow inside the macrophage. So, this is for initial part of their growth is fine, because As long as they are inside the macrophage, our immune system cannot see them, but eventually they are visible and Th1 response is there to kill them. But in case of Plasmodium falciparum, okay, I am telling how, how the pathogens are smart and clever to bypass the immune system. So, the malaria parasite grow inside the RBC, which neither express MHC1 nor express MHC2. So, while they are growing inside, no way their antigen will be presented on the surface of RBC, and as long as they are not presented by MHC1 or MHC2, either one, immune system cannot see them, T cell cannot see them, right. If T cell cannot see them, even B cell see them, can't do much. So, that way they are very, very smart parasite. But smart parasite, actually parasitism, we should not blame them, we should blame us, actually our evolution rather, because you see they are all parasite. If I talk the Plasmodium falciparum, there is a protozoa, single cell eukaryotic system, right. So, they evolved long before the human evolved. So, they are surviving. So, it is not that they grow, they came to earth to infect us, they were there, we came later, and unfortunately we are the host. Okay. So, that is why immune system is very difficult to find generally most of the intracellular parasite, particularly this kind of parasite which grow inside RBC are even tough. So, you know the cycle of the parasite, particularly the Plasmodium. So, they infect one RBC, grow inside, then they burst, many parasite particle come out and then infect multiple new parasites, just like virus lysis. Okay. So, only that period of time. So, when they are inside the RBC, nothing happen, we are, if someone is infected, then normal. As soon as they burst, the fever come with chill, right, severe chilling, and the fever started and very particular time. So, this is the symptom of malaria parasite, because you see a fever in a regular interval 12 hours or 24 hours, that time actually they burst, and as long as they are outside, the fever is there, whenever they found new host, they enter, again they are secure and immune system cannot see. So, why I am telling all this thing in immunology, because we have to know the disease also if I want to buy immune system or vaccine to protect us. So, if we have to understand their life cycle, we have to know what is the time span they are staying outside. This is the only time to attack them by immune system, right. So, even if we develop a vaccine, we have to be very careful to design that should be more efficient, and they should find that form of, because in parasite there are, this particular parasite Plasmodium, there are two forms, sporozoite and merozoite. So, that particular time they have to either we have to block the attachment of parasite with a new RBC, so that they infect, but they cannot propagate, or we have to kill them or agglutinate them or help the macrophage to phagocytose them, right. So, this is, so whenever definitely we are discussing the immune system or immunology, but what is the application of this immunology? So, we have to understand immune system, we have to understand the different disease, different pathogens, whether it is a bacteria or virus, their life cycle, their strategy, how they invade their at molecular level, then only we can different. Besides this, they also have another strategy of the malaria parasite, which is very common to many other parasites or infectious agent. They have a surface protein, okay, it is called circumsporozoite protein, which is a variety. There are at least 200 different genes, and every time they express a new one. So, even if you introduce one antibody which is specific to one circumsporozoite protein, they may not be active for the next one or next one, they have a multiple copy. So, they express randomly, and in one particular cell they have only one type. So, this is another variety. You have heard like virus is changing their surface protein, that is why antibody is not or vaccine is not working, but malaria is like that. So, all these parameters of the infectious agent we have to know in one hand, another hand we have to know how immune system work and how we can manipulate and put together if we can manage or manipulate the immune system, then only we can be successful in eradicating the different disease or development of different vaccines. Okay. So, that way, so this distribution of MHC is very important and will tell us what is their role in immune system. Okay. So, we will still continue the different few things are still there to discuss about MHC, what is they are doing, and see you in the next class.